Okay, I want to go over Lab Station 2 really quick so you have a reference in case you need it and I'm not there. So for Lab Station 2, you're going to be looking at plant growth and mitosis. You can click your gizmo here or here, right, is also the gizmo. This is what it looks like. It should look like the one with the little cells all over the place and cell division. So first thing you're going to do is read your instructions over here. Okay, it says, today you will observe the cells of two parts of a plant, the root and the leaf. You will determine which is undergoing the greatest amount of growth by observing how many cells are in mitosis and how many are not. In the diagram to the right, you see plant cells taken from a sample. When the nucleus is present, we say the cell is in interphase and not preparing to divide. If chromosomes are visible as dark lines, we say the cell is in mitosis and preparing to divide. Mitosis consists of specific phases that occur in specific order. Prophase is the first phase of mitosis where chromosomes condense and become visible. Metaphase involves chromosomes moving to the center of the cell. Anaphase is where the chromosomes are equally pulled to either side of the cell. Finally, telophase is where the chromosomes bunch up at either end of the cell and begin to unravel into their previous state. Cytokinesis then occurs where the whole cell divides between the newly formed nuclei. We also call this PMAT for prophase, metaphase, anaphase, and telophase. So that's what you're looking at is a bunch of cells. Some of them are in mitosis and some of them are not, right? We see that these are non-dividing cells. These are actively dividing cells. Okay, the ones that look like they're not dividing have a nice nucleus. They have dark spots in there called the nucleolus. These have visible chromosomes, which means basically condensed DNA. DNA is usually invisible to the eye, except when the cell is dividing. It becomes really thick. It bunches together. We can see it. Looks like pasta or noodles, right, in the cell. Okay, so that's basic. That's the point of the lab that you're doing, is to visualize these cells undergoing division or not, and the different structures inside, and make a decision. Is the cell dividing or is it not? And we're also going to look at two parts of the plant, the leaf and the root, and see which one has more division going on and why we might think that. So first thing you're going to do is on your gizmos, you're going to open it. All right, and you're going to apply. It doesn't matter. The longer you set your cell cycle length, the longer the simulation is going to take. The less is less. You don't want it super fast or slow. You can put magnify on and press play, and you're just observing. Right? We see one cell become two, and we can see when it's doing that, the cell actually looks different inside when it's actually dividing. Right? We can see the structure is doing something else when it's undergoing division. And that's all you're doing is taking observations. And you're going to put that over here. Okay? Make observations of the simulation. Over here, we have a picture of each of the major phases of cell division. And you're going to do a summary. So you're going to click over here, interphase, prophase, metaphase, anaphase, telophase. All right, and read about it and type a sentence or two. Don't need all that information. You just have to have a general understanding of what's going on in the cell and why it might look like that. So you don't have to put this whole paragraph in there, just maybe the first one or two sentences, so that we understand in general the flow of steps that occur for one cell to become two. After that, we're going to actually do some observations. So you're going to observe a root tip. So we see here, this looks like, right, if you were to zoom in on a root on a plant, up here is what we call the mitotic zone. This is where the cells are growing. Just think of a plant, how the roots all spread down like fingers. And if we zoom in on a microscope, this is what it looks like. And we can see that some cells are not dividing and some cells are, right? We can see those visible chromosomes in some of those cells, while in others we can't. So what you're going to do is fill in this chart here, actually counting how many cells are in each of the phases. All right, so up here, what parts of the cell can you see? So that's already labeled for you, right? We can see the cytoplasm, the nucleus, chromosomes, and the cell wall. Estimated number of cells, so you got to count them. You know, I don't know if you want to put in present mode or if you have an annotating feature with like a pen that might help you, you know, be able to keep track of them. You can use your finger, but you want to count how many total cells there are in the root tip. Then you're going to go down here and figure out how many cells are in each phase. So interphase. If you don't remember what cells in interphase look like, go back to this photo, right? And that's what an interphase cell looks like. So then you're going to come back to the photo here, right, and count. Most of your cells are going to be in interphase. They're not dividing. Then you're going to look for prophase. Again, look over here, prophase. Kind of looks like interphase, except it's not such a nice circle, and we can see those chromosomes, right, the noodle-looking structures in there. This one's a good example of a prophase. Right, so you're going to count how many in prophase. How many in metaphase? Again, go back to your photo. Look for cells in metaphase. Looks like everything's bunched in the middle over there. Do we see anything? One should jump out right away, right? That one looks very metaphasey. So you're going to put your numbers in here, and then you're going to find the percent. I put the equation for percent over here. So you're going to take your number of cells that are in your phase, divide it by your total cells, which you did up here, times by 100, and that will give you your percent. Remember, your percent should always be less than 100. That's how you know you did it right. Okay, and then answer yes or no, whether your cells were dividing or not. And when you're done with that, you're going to click on your formative. Right, and complete your formative for this section. So you're going to just use the picture here and answer the questions, right, using those two photos up there. Think about this one's an open-ended one. Why is one plant structure growing more than the other? Roots versus leaves. Here we have a zoomed in of a plant leaf versus root hairs. So again, this is the top part of the plant, that's green. This is the underground part, right? That's the roots. So you want to see which one is missing, so compare the two. Root hairs, even though they're plant cells, are missing one important organelle that a leaf has. And then you want to think about why root hairs might not need that organelle, what the function of that missing organelle is, and why a root hair will not need it. And when you're done, submit, and you're all done station 2.